Now let's concentrate about our last topic of this unit, Jak-Stat pathway. Kinase activation is done. Activated kinase phosphorylate receptors and provide phosphotyrosine binding site to the receptor. In this phosphotyrosine binding site, which provided by this activated kinase to the receptor, this phosphotyrosine binding site recruitment of downstream signaling molecule. On the other hand, the non-receptor protein tyrosine kinase, which are associated with cytokine receptor, there are of two categories. Member of SRC family. This consists of SRC and closely related proteins. And the second family, Jak family. Full form of Jak is Janus kinase, non-receptor protein tyrosine kinase. This is our point of interest. Members of the Jak family appears to be universally required for signaling from cytokine receptor. Now let's concentrate about Jak-Stat. Jak kinases. From the characteristics presence of two kinase domains, and it is characteristics of their family. Two kinase domains in each molecule. Several members of this family are known: Jak1, 2, 3, each associated with specific set of cytokine receptor. All have separate set of cytokine receptor. Jak1, 2, 3. The Jak kinases are tyrosine kinases whose major substrates are transcription factors called STAT. Kinase, this is an enzyme. Major substrate of this enzyme are transcription factors called STAT, and full form of this STAT is signal transducers and activators of transcription. Full form of STAT is signal transducers and activators of transcription, STAT. So what are the STATs basically? STATs are major substrate of tyrosine kinases and major substrate and transcription factor. STAT is nothing but transcription factor and substrate of tyrosine kinase, and tyrosine kinase basically Jak kinases are tyrosine kinase and which have two kinase domains each molecule. And there are also several types of STATs. So many types. Each STAT phosphorylated by a particular set of Janus kinases or JAKs, and all STAT protein contains an N-terminal SH2 domain, and through this domain it binds to phosphotyrosine in the receptor cytosolic domain. A central DNA binding domain, a C-terminal domain with a critical tyrosine residues. So STAT has N-terminal SH2 domain through which it can bind with phosphodieldosine in the receptor cytosolic domain, a central DNA binding domain, and C-terminal domain with a critical tyrosine residues. It's all about STAT. Now, once a STAT binds to receptor, then what happens? The C-terminal tyrosine is phosphorylated by an associated Jak-Stat. Then what happens? STAT dissociate from receptor. No more bind with receptor. Then form either homodimer or heterodimer. When STAT bind with a receptor, then what happens? C-terminal tyrosine is phosphorylated by an associated Jak, and after that STAT dissociated from that receptor and form either homodimer or heterodimer. The basic dimerization, it is a reciprocal interaction between and SH2 domain in one subunit and a phosphorylated tyrosine in the other subunit. Form either homodimer and heterodimer, and dimerization occur between. Dimerization is nothing but a reciprocal interaction between SH2 domain in one subunit and phosphorylated tyrosine in the other subunit. Finally, the STAT dimer move into nucleus, then bind to specific enhancer sequences calling target genes. This is the ultimate target of STAT. STAT dimer move into nucleus, and there inside nucleus they can bind to specific enhancer sequences of target genes. So this is all about Jak-Stat pathway, which one is very much important. And now let's concentrate without figure description, without drawing this pathway, all of this pathway, no one is completely completed without figure. So let's concentrate about Jak-Stat pathway figure. Figure description is much more necessary. Okay, now let's draw figures. Like all other diagrams, this is plasma membrane, and you should draw like this fashion throughout plasma membrane. PLP layer. This is PLP layer of PM, plasma membrane. This is extracellular space. This is cytosol. Now let's draw the monomer prior to ligand binding. Ligand binding domain, cytosolic domain. Clear? And ligand is coming from this extracellular environment. Ligand not attached. This is normal monomer condition. After that, what happens? Ligand is coming and dimer will form. Ligand is coming and dimer will form. Here phosphorylated phosphorylated, okay, and ligand is bound here. Dimerization. Next, what happens? Again, draw. Here, Jak kinase it is attached. These two are Jak kinases. They are cross phosphorylated each other. It gives phosphate to this Janus kinase and this Janus kinase give phosphate to this. It's already phosphorylated and activated JAKs phosphorylated. Association with STAT. P here P now STAT attachment. Then phosphorylation of STAT occurs. Enters into nucleus. Step by step Jak-Stat pathway. Let's have summarize this figure. This is plasma membrane, protein lipid protein layer, external environment from which ligand is coming, and this is cytosolic environment. These are ligands are coming. The first step monomer, second step dimerization, third step attachment. These are all Jak. These are JAKs. Jak attachment. They can cross phosphorylate each other. This is cross phosphorylate. Activated JAKs here phosphorylate receptor on tyrosine. Next, STAT attachment, phosphorylation of STAT, enter into nucleus, then bind to DNA, bind to DNA and other gene regulatory proteins. So this is how many types of phosphorylations are done. First of all, at the time of dimerization, autophosphorylation is done. Point number one phosphorylation. This phosphorylation is autophosphorylation fast. Okay. Then after Jak attachment, cross phosphorylation. Point number 2. Then phosphorylate, then Jak phosphorylate tyrosine residue. Number 3. Phosphorylation. Okay, then stat attachment occur, and here phosphorylation of stat occur. Total 4 times phosphorylations are done in the Jak-Stat pathway. And finally, this is all in a nutshell, in detail, the very important Jak-Stat pathway. So by completing this Jak-Stat pathway, unit 7 cell signaling is being completed successfully under NEP curriculum of zoology major provided by University of Calcutta, applied from this year 2023. Hope you have all enjoyed this session very well. So students, stay tuned, stay connected.